This is Ken Morris, and we're going to talk a little bit about some of the aspects of pharmaceutical equivalence in the context of the science of equivalence. These are concepts that were largely proffered by FDA folks, but formulation and pre-formulation and development and analytical scientists as well. These are discussion points and largely my interpretation of much of this. I formerly was a university professor at Long Island University, and before that the University of Hawaii at Hilo, and before that Purdue University. And what we'll do today is just talk about a few aspects of this that should cast some light on the relevance of not only pharmaceutical equivalence as a concept, but the mechanisms by which pharmaceutical equivalence may or may not be established. The sort of open questions are, what is pharmaceutical equivalence, which everybody has an opinion. It's sort of like art. You may not know what good art is, but you know when you see it. And the other questions follow, of course, which is why is pharmaceutical equivalence critical to dosage form design and manufacture between brands, generics, or across the board. The linkages between pharmaceutical equivalence and therapeutic equivalence, including the role of GMP, which is often sidelined in this discussion, and some of the analytical challenges for establishing pharmaceutical equivalence that stem from some of the vagaries in the definitions that are available. And finally, some regulatory opportunities in establishing pharmaceutical equivalence. And basically what we'll discuss, or the context of what we'll discuss, is the fact that therapeutic equivalence is the ultimate goal between either new dosage form designs or dosage form changes that occur during the development of a new drug product. However, that is often based on the bioequivalence, which, as we'll see, must be preceded by pharmaceutical equivalence and coupled with GMP. So we'll get back to that fairly quickly, but that's the context. If you look at the Orange Book, what you notice in the Orange Book is all the approved drugs, all the FDA-approved drugs, the information on it. And if you look at their definitions, that includes the approved drug with therapeutic equivalence evaluations, you see a couple of really interesting things. First, therapeutic equivalence of approved drug products are to be considered so if they are pharmaceutically equivalent and bioequivalence has been demonstrated. And then it's assumed that they will have the same clinical effect and safety profile. So let's drill down a little bit into that and see what they say about pharmaceutical equivalence. So pharmaceutical equivalence are drug products in identical dosage forms and routes of administration with identical amounts of the identical active drug ingredient, which sounds fine, except the definition of what identical dosage forms is quite different than I suspect most pharmaceutical scientists not involved on the clinical side would deem as reasonable. And that is that in their definition of identical dosage form, what it says is that they do not necessarily contain the same inactive ingredients. So I doubt any pharmaceutical scientist involved in dosage form design or development or analysis would find that to be the case. And also, if you look further, what they say is, in addition to meeting the compendial agreements, criteria rather, they can differ in the characteristics such as shape, scoring, configuration, release mechanism, packaging, and excipients. So I think you can see why there's a fairly large gap between what those of us on the development side would call pharmaceutical equivalence and on the from the clinical side, where understandably the criteria are do you give it by the same route of administration? Is it the same drug and the same strength? And is it supposedly similar in terms of the way its bioavailability is expressed? Pharmaceutical alternatives, which much of this would fall into under my definition of pharmaceutical equivalence, have all of the other requirements, but are not necessarily the same drug in the same amount or dosage form or even the same salt or ester. So they all have to meet their own respective compendial criteria, but nonetheless, I think you can see that there's a fairly large difference in what we call pharmaceutical equivalence and what is contained in the definitions. Some of that is because it's very difficult to establish what pharmaceutical equivalence is, even for those of us who have some relatively cohesive idea of what that entails. Now, the problem with not being able to accurately identify and demonstrate pharmaceutical equivalence so that you can have an equivalent is that the basis for therapeutic equivalences backs up to bioequivalence. And bioequivalence, however, is very limited in scope. It's all low variability, normal healthy subjects, and relies on a clear definition of pharmaceutical equivalence. But if you're not pharmaceutically equivalent, and you go into the bioequivalent studies, and there's variation that occurs either before or after that in the dosage form itself, there's no real way to have confidence that that's anything but a snapshot of the development, the product's level of development. So relying on bioequivalence in the absence of pharmaceutical equivalence is really a no-starter. And in fact, even with pharmaceutical equivalence, it has to be coupled with good GMPs for reasons we'll talk about. So you really need to have meaningful specifications for a product set before it goes into the clinic. And this is true whether you're talking about across generic sponsors or during a development campaign from a branded company of a new compound, when you make changes and do bioequivalence studies, if they really are representative of the product that ultimately goes to the patient. As is often said, you'll never know less about your product than the day it's approved and distributed. So we need to move pharmaceutical equivalence to the latest century, and the idea is that new drugs get the right CQAs, which are in the context of considering the patient needs and failure modes, and that the same CQAs and lot-to-lot variability apply for generics. Much of these concepts are contained in the reference here from Ejaz Hussain and some of us in NIPT. Now, people have, as I said, from FDA have been dealing with this for some time because it's obviously part and parcel of what must be taken into account for reviewing. Rob Lineberger, who's been a thought leader for many years in FDA, in 2014 had tried to zero in a bit more precisely and had come up with definitions of equivalence based on a three-tiered system, Q1, 2, and 3, where he would classify the product similarity based on whether it had the same components, and then whether the same components were in the same concentration, and finally the same components, the same concentration with the same microstructure, essentially. That's the arrangement of matter. There's a word cut off here. And you can see, although Rob had zeroed in on it, there's still some vagary because in the Orange Book definition, the same components may actually be referring to excipients with the same function, but not necessarily the same function, same excipient. Not that he wasn't aware of that, but just so you know, there's just room that there's still things that have to be much more clearly defined than they currently are. So if we look at this in general, and at this point I'll pause and bring up another program. So here we can map the pharmaceutical equivalence criteria to some degree based on, starting with Rob Leinberger's formalism, the Q1, 2, and 3. And you'll notice we've added one extra Q that we'll talk about a little bit. And again, this is somewhat speculative, but hopefully will add to the conversation. And if you expand these into what would be logical for those of us who are familiar with development, and relevant to those of us who end up having to do the analyses on these products, you can see that at each level there are very similar looking categories. You have to have the API equivalence, excipients equivalence, and then use tests. But what I'll show very briefly here is that the granularity becomes there's more granularity as you go to the higher Qs, and in some cases the requirements change slightly as you go as well, based on the level of similarity. So for API, for example, even at Q1, I think we would say no matter what the official definition is, you'd like to be able to ensure that you have the same form, polymorphic form in this case, and there will be associated analytical methodologies that you will use that we're familiar with, X-ray and spectroscopy of various sorts to confirm that and maybe quantify them as necessary, and also determine their behavior. And then when it comes to purities and impurities, you'll also have the requirements for identifying the impurities, either those that are as in addition to those that are specified by USP, any others that you may think are relevant, which may include solid state impurities, if you will. And in excipients, keeping in mind that at Q1, you don't necessarily have to have the same excipients, but you do apparently have to have excipients with the same function. And in that case, what we're saying is, well, you need to have some critical quality attributes. I'm focusing on solid oral dosage forms and small molecules at this point, but similar maps can be generated for more specific products as needed. So for those CQAs, you may have micromeridics, that is the particle size, surface area, porosity, and there are techniques, of course, associated with those as well. And impurities again, but now in excipients, not necessarily solid state impurities. And then also, the mechanical and other properties that actually matter for making products not only at the kilo level, but at large scale. What's interesting, however, though, is that when you look at excipients in terms of their variability, because you're not limited to one excipient per function in Q1, it's actually more burdensome than it may be in later stages, because now, in addition to looking at the consistency of an excipient across different lots that you're using, you also have to be able to determine the variability across different excipients with the same function. And so here, CQAs may vary depending on whether or not it's a molecular weight or particle size, whatever it is. So that becomes a relatively burdensome methodology to have to institute if you haven't decided on it. And of course, it provides FDA with challenges when they're doing their reviews. When you get to Q2, there the excipient variability is actually going to be just the lot-to-lot variability, or the consistency of the excipient from your source for whichever excipients you've chosen. So as you go further along, some things drop out, like the inter excipient variability, but other things pick up, and we'll see that more as we get into the use test, which is what I was going to talk about next. If you look at the use tests across the Qs, and this is now again focused mainly on solid oral dosage forms, you may have release profiles, macroscopic content uniformity. By that I mean the content uniformity across dosage units. There are various techniques that we've done, HPLC, and then I put examples of XRF and other things that we've been discussing in the context of these talks with Simon Bates, who's the diffraction physicist who's been involved with all of the solid state characterization in the universities that I've been in, as well as many of your companies and universities. So with use tests, though, you have now again an inter and intra source excipient variability that you have to deal with. That is, you need lot to lot, but you also need that for each excipient that you've chosen, and chemical reactivity. So the reactivity can take many forms, as we know, but this is just an example point. As you go to Q2, things look pretty similar. The place where I would draw some distinctions are that here, as we mentioned earlier, you now have an intra-source of excipient variability, lot to lot, if you will, that has to be established. What I would say is that the addition of micro content uniformity could come into play here, and it definitely will when you get into Q3. So in Q3, what we see is that we have above this in the hierarchy now, we have an additional constraint. This is what Rob Lineberger called microstructure. And so here you also have the additional possibility of looking at the distribution of your API within your dosage form. Is it's associated with other components in the formulation more intimately in one than the other. Also, is the distribution of crystal form or amorphous or disorder in the API varied, and does the density vary as you go along? Or, as you'll see in one of the examples, does coding vary because of the variability in the process itself? So in the use testing, where you have all the other requirements again, and I haven't expanded on a lot of these, but here a little bit, you could have whole dosage unit content uniformity. You can have the CT spatial density, coding variability, microscale. It would be API and excipient distribution, X-ray used in the context of spatial form determination, and then again the ones that are going to follow along as we go. And finally, what I would just add is that this is one of ours that we've added as a group from I should say as a section from our group discussions, is Q4, which is trying to tie the variation that we see when we do all of the other levels of analysis to something that is relevant to actual dosage form performance in vivo. Not that we're doing in vivo work, but something to link it. So in particular, what we've talked about, and there's many PK programs, but SimSip is the one that we used most continually at Long Island University when I was there in David Taft's hands. So there is SimSip modeling that can be done that will allow you to input the variation from use tests, so dissolution, for example. But it may be other things. It can be characteristics of products and solubility, etc. That lets you model what the PK will look like when you change it within some bounds. So in a sense, it's getting towards the design space concepts. And of course, this can include things like enzymatic susceptibility of your compound, and you can model the bioavailability to some degree using the input from the Q3, and then linking the use test to pharmacokinetics. There again, you can input all of the behaviors that you're getting from the use tests here as input into the PK modeling program to see, to sort of scope the level of impact it may or may not have. So there's two levels of interest here. One is our interest in meeting compendial specifications as well as the consistency of the manufacturing and in vitro performance. And then the other is there a link to something that will potentially impact on patient-based failure modes? So if we return to the PowerPoints. From here, let's talk a little bit about some implications. So I'll sort of start from the large scale. So this is a study that was done many years ago in 2001 by G.K. Raju when he was involved with the CAMP consortium between Purdue and MIT. CAMP was the Consortium for Advanced Manufacturing Pharmaceuticals that Steve Byrne and Charlie Cooney headed. And they'd done a study with eight of the companies, the largest companies in the world at that time. I'm not sure Pfizer was one of the largest at that time, it wasn't involved, and looked at their products, and basically, conceptually, it was going to the plant manager and saying, how long does it take you to get a production batch through? They would tell you, and then you would look at the data, you being GK and his team, would look at the data and say, how long did it actually take? So this is done for many, but this is just a detailed analysis of 2 products, but it's pretty representative of what they found across the board, was that the estimate that the plant managers made were in the neighborhood of 25 days. The average, it turned out, was 95 days. Now, this for these 2, so it varied, but it was in that neighborhood, a factor of 3 different, let's say, or 4 different. And notice the standard deviation was greater than the average, and the exceptions increased cycle time by about 50% and increased the variability by over 100%. So it's low capacity of utilization. And you can see that graphically here, where you have sort of an idea of what the average throughput was thought to be versus what it was. So these were all to do with exceptions, and some of the ones that took longer without exceptions were just because of delay in getting material, perhaps. But the OOS investigations may never resolve. I've been involved with them that have been going on for a year or 2 years and never resolve. And that's because it's hard to back into what was the root cause of a problem. And if you don't really understand what the pharmaceutical equivalence is, it's really hard to figure out where things might have gone wrong. And the other element that enters into this is that every time you do an OOS or something, the QC testing times are really high. And these are just different case studies. These are overall cycle time components, processing time in blue and QC testing times. So you can see that depending on what the tests are, it can really take a long time. So any type of investigation that propagates more testing is compounded by the cycle time increase due to the testing. So now let's look at a couple case studies specifically on products. This is a case by mesalamine, on mesalamine, and it's the subject of a guidance and a paper by Ajaz Hussain. I've got the reference shown later. For some reason it's not here. But what they found is there's really high variability in dissolution for this product, and if you look at a controlled release coating product, what you know is that there's going to be some variation in coating. So usually you'll add coating up to a certain weight gain in most cases. And what they found was that no surprise that as a function of pH, there were pHs where it was instantaneous, but as you went to more discriminating pHs, you saw very large differences. So even though the overall coating criterion in manufacturing was met, when they had done terahertz analysis of the coating thicknesses and compared them to the mean dissolution times, what they found is that the coating thickness was not a huge correlation, but a pretty good correlation, that the coating thickness, the average coating thickness, was responsible for this huge variation. And the message here that comes out of the paper by Ajaz Hussain is essentially that you can adherence to the principles of ICH Q8 and GMPs are not always the same as compliance with the regulations or the guidance. So the USP may say that you have to have a Q of not more than 20% in 30 minutes and not more than 80% in 60 minutes or whatever it is. But that doesn't necessarily tell the whole story. And there's another case with methylphenidate that's pretty interesting that has a similar message that you may be aware of. And this led to a guidance which basically said that you should be doing more discriminating dissolution testing. And it just comes back to this idea that it's known that the dissolution is dependent on coating thickness. So what good is bioequivalence if you don't know what the pharmaceutical equivalence is? If you don't know what the variability in the coating thickness is, it's very hard to put a lot of confidence in a relatively small bioequivalence study, as most of us are familiar with. And the last example is sodium levothyroxine, and there's been a lot of work on this. This is just a real quick review of the history. It was synthetic levo was first available in 1955, and from 82 to 97, these super and subpotent problems were just constantly reported. It's the most recalled drug in history by any measure, whether numbers or numbers of units or numbers of recalls. And then because it was until 1977, GRAS, no review process was required. And the problem was, and this came out, Eric Duffy had presented this at one of our ACPS, this is the Pharmaceutical Science Advisory Committee meetings at FDA, and showed that as you went from manufacturer to manufacturer, the variation in the stability was just all over the place. And nobody had really resolved that to any satisfactory level until just recently. And in 2001, when levo was declared a new drug, it of course has to go through more rigorous approval process. And we at that committee, we tightened the specifications to some degree, but this is sort of a Band-Aid on the problem. It wasn't identifying the root cause. So because it's such a narrow therapeutic index and low-dose drug, it was very problematic to establish what a pharmaceutical equivalent system was and what the possible failure modes, that is dosage form failure modes, there were in terms of the stability itself. So as we said, it's the most recalled drug in history. And the paper by Harsh Shah here actually details this in much more detail. So it remained, as we said, unclear for a long time, and the fact that the very low dose, that is 25 to 300 micrograms in a 200 milligram tablet, with a variation in dose of about 12 micrograms between doses in some cases, very hard for the doctors to titrate this. People would get titrated on one, switch to another, and they would have different reactions. And even amongst the ones that they were titrated on, especially before the specifications were tightened, they would, even with the same compound, depending on its age, could still be within its shelf life and be sub or super potent. So we started looking at this from the standpoint of the structure. Actually, at the FDA meeting, this was first pulled up, and this is the Cambridge database single crystal structure. And what you notice is this falls into a class of behaviors that Steve Byrne had identified many years ago in his book. And if you look at the structure, you can see that along certain axes in the crystal lattice, these are 4 unit cells together, that the sodium and water molecules are sort of lined up in these channels. And this is just the unit cell dimensions, because we'll show that these cell dimensions can change depending on the exposure. And if you look at the powder diffraction pattern, you'll notice that when you treat the material differently as a function of exposure to humidity and temperature, that these channels seem to be labile. And you can sort of see this distance here is this represented by this peak in the powder diffraction pattern. And if you follow this as a function of conditions, you can see these changes occur. And the way that this was best followed, this was from Triclinic Labs when Simon Bates was there. And this is the vertical transmission PXRD. You can see that with the 001 plane, that there is a shift as you go from lower to higher temperature, which corresponds to dehydration. But up until a certain temperature, you can also see that the packing motif isn't changing, so that the moisture seems to be coming out without changing the packing motif very significantly. And that was key to some of the behavior that we later elucidated in the paper. Also, as we say here, there is some disorder that occurs. You can see it here, but you can also see the fall off here. All of that gives rise to the idea that if you were to look at this structure and schematically, we did this schematically first before we actually had collected data. This was the original hypothesis, that if you were to subject it to low RH or high temperature, that you would contract the lattice, and hence the change in the D-spacing and the peak at the 2 theta value at the peak that we were monitoring. So once you dehydrate it, you can rehydrate it by changing the conditions, and it'll expand back, and that's why the retention of the packing motif up to a point is important. So once you evacuate the channels, oxygen can get in. If it's disordered, the oxygen may have more accessibility to it. And if you look at the reactive sites, they do seem to correspond with those positions. And so let's put that together. How does the solid state change really relate to the chemical stability? So here's the effect of RH and temperature. This may be some work that we had done at the University of Hawaii with Maz and Hamad. This is the percent of levothyroxine sodium remaining. And the degradation products for this, first it loses an iodine, goes from T4 to T3, but it's a whole cascade of reaction pathways. And you can see that if you look at, at low humidity and high temperature, that you get very rapid degradation. Low humidity and room temperature, you'll get some significant degradation. As long as you keep it hydrated, not independent of temperature, but not nearly as dependent on temperature, you can retain the stability. So the dehydration precedes degradation. So these falls into that category that Steve Byrne identified of degradation that is a function of it being preceded by dehydration. And if you look at the effect of oxygen and temperature, you put the other piece of the puzzle to bed, and that is that when you have full oxygenation and dehydration, then you'll get a lot of degradation. And if you have room temperature and with O2, you'll get some degradation. And if you have no oxygen, even when you dehydrate it, it will remain relatively stable. So we know that it's an oxidative degradation. So the point here is that in principle, this could have been known a long time ago, but clearly, depending on what the manufacturing process is and what state it leaves the material in, it will affect, at least potentially, what the stability is. Similarly, there's work by others that show that different excipients will also have stabilizing or destabilizing effects. But it all comes down to saying, is it pharmaceutically equivalent with respect to relevant metrics, that is relevant to the output that we're really interested in. So, to wrap this up, if we look at those same questions, well, what is pharmaceutical equivalence? The short answer, of course, that legally it depends, but I would say scientifically it has to be much more like Q3 than it does by like Q2 or Q1, unless the specific product justifies being able to relax those constraints. So there's no one right answer. However, the right answer is always to assess the critical quality attributes and criteria of utility of the API and the dosage form with respect to its routes of administration and exposures to decide what the equivalence should be for that product. But in general, it's better to start with Q3 and work backwards than it is to assume Q1 and get burnt. Why is it so critical to dosage form design? I think we know that because if you don't, then by the time you get to your bioequivalence study, then you really are in the dark. And maybe your bioequivalence study is relevant and maybe it's not. And with respect to brand or generic, I think we've addressed that in that whenever you're changing anything, you have to at least assess whether there's a degree of equivalence that is necessary to be maintained. And if not, then if you don't do that, then again, you're sort of trusting your luck. The linkages between the PE and therapeutic equivalence really comes down to either anticipating it by modeling or by having the clinical data to back that up. There are within the cadre and the sort of range of pharmacokinetic software, there are many aspects and many capabilities to try to anticipate issues. The drug-like properties concept of whether or not something is an efflux transporter substrate, for example. These are all things that could help. And then the key analytical challenges, I think, by and large, and this is something that Ijaz Hussain and Steve Byrne and others have been preaching for many years, is that the solid state and solid state analysis is really our Achilles heel. What constitutes validating a method for HPLC or a solution technique is far different than what has been done with respect to solid state. And this is partially because of the lack of attention of funding agencies, but it's also because it's just more difficult. And Simon Bates has made a career out of looking at very exactly those issues and has made very significant strides, as have others. Eric Munson at Purdue with solid state NMR. There's a rich literature on it. It's not been very thoroughly integrated into what we have. In terms of regulatory opportunities, basically the idea is that the more you can generate the information on pharmaceutical equivalence, the smoother the review process has to go with respect to the CMC, of course. This is just, the FDA often doesn't get all of the data you have for one reason or another. On the other hand, this kind of data, these data would undoubtedly make the job much more meaningful and hopefully faster with less cycles. So, again, the message here is that pharmaceutical equivalence is actually the foundation that you need to establish for bioequivalence, which then has to be integrated with good manufacturing practices to ensure the identical or at least the very similar variability of your routinely produced dosage form at full scale, and then, and only then, can you be assured of therapeutic equivalence. So, thank you, and goodbye.